use that energy in that bond to make an ATP. In fact, in this case, it makes a GTP, but then GTP is easily converted to ATP by different sets of enzymes at no free energy cost. So that gives us succinate, and then we have a three-step sequence: a dehydrogenase, a hydration, addition of water, and another dehydrogenase. And this is important to make note of: FADH2, addition of water, and NADH being generated. So that three-step sequence by which we go from CH2 to C double bond O. So we've oxidized the substrate in 2 successive steps. We've gotten 2 redox cofactors out of it, 2 successive 2-electron transfers. That set of 3 steps is the same as the set of 3 steps that will oxidize a similar structure with CH2 groups to carbonyl in fatty acid breakdown. Fatty acids, of course, lots of CH2s in a row, and they're going to go through an oxidation process that has, as its core, is basically these 3 steps of the TCA cycle. So that's 6, 7, 8. Okay, so let's look at the enzymes. Say something about these enzymes. Citrate synthase is a Claisen condensation, a carbon-carbon bond formation reaction that's driven by that release of free energy in the high-energy thioester bond. And the overall delta G, the standard state of this, is similar to what it is to what you can get out of that thioester bond. Oxaloacetate concentration in cells is low. So Q in the cell, the delta G in the cell, delta G equals delta G 0 prime plus RT ln Q. The actual delta G in the cell will not be as irreversible as this, but it's still irreversible enough to be considered irreversible. In other words, when we look at the overall TCA cycle, we don't have an arrow going backwards here. It's also an induced fit enzyme. There's some discussion about that in the text. Not so crucial for our purposes. And so the product of this is condensation. Here's your 2 carbons from acetyl-CoA, and here's citrate, which is formed. So you can look at citrate as kind of like a row of 3 carbons, each of which has a carboxylate attached to it, and the central one of which also has an OH instead of a hydrogen. If that helps you remember the structure of it, but you don't have to, this is not one you have to really memorize. Okay. So again, we've done this because the cell is finding it advantageous to incorporate the glucose breakdown product from glycolysis and PDH into another metabolite, so that that metabolite can form part of the metabolic hub to organize a lot of metabolic reactions in the cell, from which the energy of glucose can still be extracted. And you can still decarboxylate this molecule, which is what's going to happen in a couple of steps very soon, but you haven't basically broken it up to methane and lost your ability to make the hub. Yeah.
How easy or difficult is it to make something like a triglyceride out of one of these? Or is that totally off base?
No. I mean, glycerol skeleton, you don't have all the carboxyls on it, but, if you were to oxidize those carboxylate groups, then you could, I guess, get to a glycerol skeleton. Or, sorry, reduce them. There are inner conversions of fat and carbohydrate metabolism, and we'll talk about that a little bit. Yeah, but in general, that's not a transformation that is that relevant to the way biochemistry has evolved. Okay, so citrate synthase is followed by this enzyme called aconitase, which is an interesting enzyme for a couple of reasons. But my favorite part about aconitase is it has this iron sulfur cluster in it. And I don't know, have you guys seen iron sulfur clusters before? Maybe not. There is an assembly process in cells by which cysteines on certain enzymes are converted from cysteine to persulfide, which means instead of CH2SH, it goes to CH2SSH, and then that extra S gets donated into forming these clusters. So here we've got an S going directly, but here you've got another S that's not coming from a cysteine. Right? You've also got inorganic iron in the cell and proteins that are binding those. And then you have scaffolding proteins that help bring this together, assemble a cluster on a scaffolding protein, and then the scaffolding proteins target other apo enzymes like aconitase. Like an apoaconitase would be aconitase without the iron sulfur cluster, and then that cluster is delivered fully formed into the active site of aconitase, where it takes up residence near the binding site for citrate and thereby stimulates the reaction of citrate. So this happens all over the place. So there's hundreds of enzymes in the cell that have iron sulfur clusters embedded in their active sites. And the iron sulfur clusters, this is one that is Fe4S4. There are other classes of them. Looks like Fe2S2. And we'll hit some of this when we talk about oxidative phosphorylation. Usually these cofactors are used to facilitate redox reactions. So one of the irons in an FeS cofactor would go from Fe2 to Fe3, right? Accept electron, donate electron back. And in so doing, it would function as a redox cofactor, just like NAD would function as a redox cofactor, go from NAD to NADH. Right? It's just that this redox, this cube of iron sulfur would be sitting on an enzyme, taking electrons and giving them away as part of a chain of electron transfer. Right? So that's usually what iron sulfur clusters do, and we will see them doing that when we see the oxidative phosphorylation. And in fact, if you go back to the table in chapter 13, you see Fe all over the place in that table. Some of those Fe's have to do with iron sulfur clusters. But here, it's not a redox reaction, is it? I mean, citrate to isocitrate, all you've done is move around where the OH group is on the skeleton of citrate, right? So there's no redox going on, and yet there's still an iron sulfur cluster. So the only thing the iron sulfur cluster is doing is it's helping water ionize. It's providing a way to stabilize an OH minus, okay? Which is unusual, because why do you need an iron sulfur cluster for that? I mean, I could do that with a magnesium ion. And why bother assembling this iron sulfur cluster? And I don't have an answer to that. Right? It's biology. It works in mysterious ways sometimes. There's a proton. If this is citrate here, and the three central carbons of citrate are these three, it's hard to see sometimes what citrate really looks like when it's kind of drawn in certain ways. But these are the three central ones, and there's your three CO minuses around the outside. There's a base that removes a proton here, and then there's ionization of this OH minus. And in that process, you go to a negative charge on the oxygen, and so the iron part of the FeS cluster is stabilizing the O minus that comes off there. Okay. So elimination of water facilitated by one iron. When you get to this middle part, you can you resolve the cis aconitase. This is with the water removed. Then water attacks back. This time it's attacking here to form this. Water could also attack to go back this way, but thermodynamically, the isocitrate will go forward. So the concentration of it gets depleted as compared to citrate. So that helps drive the reaction forward, even though the overall standard state delta G is not favorable. Okay, so you know, just a rearrangement reaction involving elimination and readdition of water to move the OH group around. An unusual use of an iron sulfur cluster. And then we